Welcome back to vaccine design course. Now the next, under functional analysis of antigenic protein, our next step is to predict the cytotoxic T cell lymphocyte epitopes in a particular protein sequence. For that you need to go to the server named CTL PRED. Okay, so these CTL, or the cytotoxic T cell lymphocytes, they directly kill the pathogenic cells. So we are going to find out that type of epitopes. Okay, the epitopes which directly kill the pathogenic cells. Okay, so for that you need to go to this particular link and then input your sequence, make it as the FASTA sequence format, and then do the further steps. Let's go one by one. So Google CTL PRED and go to the third result, which actually goes to the help page where you can find all the information about how to submit the sequence and the prediction approaches and the interpretation of it. Please have a read at it. But here what we need is not the help button. So either go to this particular link or go directly here. Okay, so here what you need to do is need to input your sequence, which we already have. Okay, input your chain S spike glycoprotein. That's what we are going to do it as demo. So if you want to give your name of the sequence, you can give, that's all optional. Then either you can paste your sequence or again upload your sequence. Now I have uploaded in FASTA format, so it's standard sequence format. If you have removed your FASTA header, then it would be amino acids in single letter code, right? Now let's go to the prediction approaches. Here you have different predicted approaches, as you can see. So when you go to the help button, they'll mention what is the quantitative matrices based method, what is the ANN based method, SVM based. So let's go for the combined approach. Okay, combined approach, and then you have something called as display top. How many peptides do you want? So do you want only 3 peptides? So let's make from 3 to, let's say, 50. If you want 100, yeah, that's all depends on your research aim. So after that, if you want to give your cutoff values, go to the help button and see what are these cutoff scores. It's just like the threshold score that we give. So it was the combined approach. So in the combined approach, the software again says the default cutoff values, and then you need to click the run prediction method. Click the run prediction and wait for the results to come. Okay, so here the results, as you can see, they are based on the combined approach, which is, so combined approach means it's 2 algorithmic based. Okay, again read the help button. Now in results you can see color display and overlap display, where the red colored ones are the epitopes. So you can see the results in detail in tabular display. So there is color display, overlap display, where you can see the red colored ones, which are the epitopes. Now let's go to the tabular display. Okay, here in the tabular display you can see the peptide rank, start position, sequence, score, prediction, and the MHC results. Okay, or MHC restriction. So MHC restriction means it's the requirement that target protein sequence or target protein expresses MHC molecules that the T cells recognize in order for the T cell to respond to the epitope present by the target protein. So it says the first one means that's the most efficient epitope, then it goes one by one followed by the peptide rank. Okay, you can see the start position and the sequence as well. Okay, whereas since we have given it as a combined approach, that's why the score, that is the 2 algorithmic based, which is ANN and SVM based algorithmic score, is mentioned 0.93 and 0.70. This one is the first, or you could say the efficient epitope, followed by the others. So as you can see, you have, since we have displayed it as 50, there are 50 out of 50, 47 are the epitopes. Okay, and you can also see the MHC restriction sites as well. So yeah, so this is how you need to predict. So you have again 47 cytotoxic T lymphocyte epitopes. That's how you need to interpret your results. Okay, so the server that we have studied was CTL PRED, and now you know to interpret the result as well. So that's about it. Thank you.